Today's lecture we will discuss in detail all about reverse transcriptase PCR or RT-PCR. In the previous video we have seen in detail what is PCR. So reverse transcriptase or RT-PCR is another commonly used PCR type in molecular biology and is different from simple PCR. We will see how. It is a method that is used to amplify, isolate or identify a known sequence from a cell or tissue RNA library. Here RNA is first converted into complementary DNA or cDNA, and this is done using an enzyme called reverse transcriptase enzyme. So basically it is creating a stable DNA copy from less stable RNA molecule. So instead of DNA, mRNA is the starting template. The reverse transcriptases are the RNA-dependent DNA polymerases, which were initially discovered in retroviruses. And these retroviral reverse transcriptases have different structure, but share two main elements, a DNA polymerase domain and a ribonuclease H domain. So what are the key steps in the RT-PCR process? As we have discussed that in RT-PCR, RNA strand is the starting material, while the other ingredients that are required for running the RT-PCR are the primers. That can be a random primer, Oligo DT, or gene-specific primer. We will look at these different types of primer later in this video. We also need reverse transcriptase and dNTPs. Now the first step is the synthesis of DNA-RNA hybrid, and this is done by RNA-dependent DNA polymerase activity of reverse transcriptase. Additionally, we have seen that the reverse transcriptase also have an RNase H function. Now this degrades the RNA portion of the hybrid, hence resulting in the single-stranded cDNA. This single-stranded DNA molecule is then completed by DNA-dependent DNA polymerase activity of the reverse transcriptase. And as a result, there is a double-stranded cDNA molecule. Now before discussing the types of RT-PCR, let's have a look at different types of primers. Random primers, these are the short single-stranded sequence of usually 6 to 8 nucleotides, and they bind at the multiple complementary sites of all RNAs, with or without poly-A tail, for cDNA synthesis using reverse transcriptase. Oligo-DT primers. Oligo-DT primers are mostly 12 to 18 thymidine residues that binds at the poly-A tail of mRNA. It generates a full-length cDNA from poly-A tailed mRNA and is good to use if starting material is less. However, it has a disadvantage that it can only amplify a gene with a poly-A tail. Sequence-specific primers. These are specifically designed short single-stranded sequences of nucleotides that only bind to specific region of interest of RNA. RT-PCR is generally of two types based on the steps it is performed, and these are one-step PCR and the two-step PCR. In one-step PCR, reverse transcription and amplification reactions all occurs in a single tube. First, reverse transcription occurs forming cDNA, which is then amplified in a PCR process. As both reactions are conducted in a single reaction tube, sequence-specific primers are used in this type. It is easy to carry out with a less handling. Due to a reaction in a single tube, one-step PCR is generally easy to carry out as it requires less handling. It also has a disadvantage, as fewer templates per reaction mixture can be detected due to multiple chemicals in a single reaction tube. It is also less sensitive and there is an increased chance of primer-dimer and the non-specific binding. Moreover, the chances of reaction failures are comparatively high. In the two-step PCR, The reverse transcription and the amplification process all occurs in two separate tubes. In the first tube, a reverse transcription reaction takes place, yielding a cDNA. These cDNAs are then transferred to a separate tube where PCR mixture is added and the cDNAs are amplified. You can see in this type of PCR, reverse transcription occurs in presence of either random or oligo primers, while for PCR reaction, gene-specific primers are used. As two-step PCR is done in two steps, it has higher efficiency and accuracy, and the chances of primer-dimer formation is quite low. However, two-step PCR requires extra steps of optimization, thus making the whole process a bit more complex than one-step PCR. So that is all about RT-PCR. In the next video, we will see how RT-PCR is different from qPCR. So stay tuned and don't forget to subscribe my channel for more amazing videos.